Now we're going to start getting into photosynthesis. So we're going to begin by talking about the discovery of photosynthesis and how that happened. So there was some pretty intelligent experiments that were done back in the about mid of the 1700s, but it wasn't really clear how the process actually worked until much later. But there were some pretty creative experiments to start to describe how photosynthesis operates. So one of the studies took some glass jars and enclosed things in there that use oxygen. So fire uses oxygen. You need oxygen to keep a fire going. So you put a candle in a closed jar and it goes out, right? That's because it uses up all the oxygen really quickly. There's nothing left to fuel it, and it burns out. The same idea happens if you say you put a mouse in a jar. It'll live for a little bit, but if it's airtight, eventually the oxygen runs out, carbon dioxide builds up, and the mouse will die. So what they did is they put in a plant inside with the fire, and they found that the fire would keep going. It didn't go out right away, and that was they deducted because the plant is taking out the CO2 that the fire is emitting and putting oxygen into the air, and so they've started to create basically an ecosystem, right? The fire produces CO2, the plant takes it in, the plant produces oxygen, and the fire takes in the oxygen, and so the fire didn't go out right away. It lasted longer. Then they did it with the mouse, and they found the same thing. The mouse continued to live. The plant produced oxygen. The mouse used the oxygen, produced CO2, which the plant took in. And so they were able to deduce what plants were doing. That plants were emitting oxygen and taking in CO2. Pretty intelligent, simple study. Now, what they then need to do is figure out, we know what it's doing, but how do we know if it's using light, or like what's the source here of energy for the plant? And so they found these bacteria that are actually attracted to parts of the plant that are undergoing photosynthesis, right? And so what we call I can't really term for it, but bacteria that are attracted to oxygen, and since their plants are excreting oxygen from photosynthesis, the bacteria become attracted to it. And so they took this algae, Spirogyra, which is photosynthetic, and they exposed it to a gradient of light spectrums. So light exists in a spectrum depending on the wavelengths, which we'll talk about in a second. And so they exposed different parts of the algae to either red light or yellow light or green light or blue light to see where the bacteria tended to go. And you can see here on this graph that the bacteria, these red dots, went to the part of the Spirogyra that was exposed to the blue light. Very little bacteria were in the green light spectrum, and there's a lot more of it in the red light spectrum. And that matches up with what we now know about photosynthesis, is that plants don't really use green light. They're using blue lights and red lights, but not so much green. And that's why they reflect green light. That's why they look green to us, because they're not really using that light. And so they could deduce that at least Spirogyra was using the high and low wavelengths of visible light to carry out photosynthesis. And they were able to do that by looking at where the bacteria tended to cluster. So it's a pretty creative study showing Spirogyra does appear to use light and different spectrums of light, and that it seems to be taking in CO2 and out exporting oxygen. So for photosynthesis, we now know that plants take out oxygen, or breathe out oxygen, take in sunlight, take in carbon dioxide, make sugars, and then take in water from their roots. That's the basic fundamentals of photosynthesis, at least from a very broad perspective. As we look more into the cellular components, we know the chloroplast, this organelle here, similar to mitochondria, becomes very useful. And it's composed of a similar anatomy to a mitochondria. It has two layers, so the outer layer and the inner layer, and they have that layer system because they used to be prokaryotes, basically bacteria that got engulfed by some sort of a eukaryotic cell through a process called endosymbiosis, and now lives inside of those cells, in our cells and plant cells. Not chloroplast in ours, but mitochondria. That outer membrane, though, is actually like host tissue. It's more similar to the tissues of the plant than it is to the bacteria. And the inner membrane is the bacterial membrane, the chloroplast membrane. Same reason why mitochondria have two membranes. So they have this two membrane system, and then inside of there they have these stacks of little discs. Each disc is called a thylakoid. Then inside they have a disc or a chamber within the thylakoid, so the inner thylakoid space, what they call the lumen here. And then between the thylakoids and the outer membrane, so in the empty space in there, we have the stroma, basically the fluid around the thylakoids within the inner membrane space. And if you have a stack of thylakoids, you have a granum, singular, or a grana for plural. Okay, so here's the photosynthesis equation. Carbon dioxide plus water plus sunlight goes to sugar and oxygen. That should seem familiar to you because it's actually the opposite of the cellular respiration equation. Cellular respiration, we take in oxygen, we take in sugar, we convert it to water, carbon dioxide, and energy. So here the energy is sunlight and it's an input. Cellular respiration energy is the output in the form of ATP. And the sugar and oxygen, carbon dioxide and water are just on opposite sides of the equation. So from a broad perspective at least, cellular respiration and photosynthesis are opposite of each other. The actual steps to the process are not opposite of each other, but the general idea is. Okay, now photosynthesis is composed of two main cycles, what we call the light-dependent reactions, the reactions that depend on light, and the light-independent reactions, the reactions that do not depend on light. That's also part of what's called the Calvin cycle. So the Calvin cycle, also called, you might see it, the dark reactions, although that name's kind of faded out now, because it doesn't really have to be dark, it just doesn't need light. Now, the light-independent reactions are generally going to happen inside the thylakoids. So they're going to happen on the membranes and inside of the lumen of a thylakoid. The Calvin cycle is going to happen in the stroma of the chloroplast. And there's some exceptions to this, but in general. But the thylakoid is the important part for the light reaction. So these things are going to kind of go back and forth between each other, but generally the light reaction is going to produce ATP and NADPH. So just like in cellular respiration, it's producing batteries that we can then power to make something. Now, remember photosynthesis equation, the goal here is sugar. Oxygen is a byproduct, almost toxic to plants in high quantities. Carbon dioxide and water are the inputs. The goal here for photosynthesis is sugar production. So for the light reaction, you can see that they're going to make ATP and NADPH. Now, ATP here is not the goal. It will be used up. So the ATP and NADPH will go to the Calvin cycle, be used up, and then recycled. With the goal here is to make this G3P molecule, which will be used to make glucose later on. And we're going to go through each one of these in turn here. So before we get into the details of it, I want to talk a little bit about light energy. So light, you can depict as a wave. It has crests and it has troughs, so low points and high points. And the wavelength is the distance between two adjacent crests or two adjacent troughs. They're the same measurement. So if the waves are really close together, it has a short wavelength, and as you move them apart, the wavelength gets longer and longer. Now, down here, we have wavelengths, right? And so you can see short wavelengths over here with the darker color, and really long wavelengths you get over to the red color. And so the spectrum exists short to long wavelengths. Now, when we look at, this kind of flopped it here, so you can see 700 nanometer wavelengths to 400 nanometers. You can see the troughs here. So red, the wavelengths are really long. Orange are a little bit shorter. Yellow is a little bit shorter. Green shorter than that. Blue still shorter than that. Indigo and violet, and they get shorter and shorter and shorter and shorter. So the blues are shorter wavelengths. The reds are longer wavelengths. Now, when you look at energy, the blues and the shorter wavelengths are higher energy, and the reds are lower energy. And so they exist on a spectrum of energy, with the shorter wavelengths having a higher energy production, or higher energy inside of those wavelengths, inside of those photons of light. Now, plants have what we call pigments to try and capture that light. Now, the main ones are chlorophylls. Chlorophylls are the ones that give plants their green color, because they're capturing light that is not green. And in land plants, there's 4 different types, A, B, C, and D. The main ones for land plants are chlorophylls A and B, right? Those are the ones found in land plants. There's a variety of other ones. I'll pull up a chart in the next one. Carotenoids are another big one, though. This is a very diverse group. You can't characterize it as well as you can chlorophylls. but it gives a lot of color to fruits and seeds and different things like that. But in a time when there's a lot of light on a leaf, sometimes it can actually be damaging to the leaf, and carotenoids can help capture some of that light and divert the energy away. So they perform a variety of functions. Now, this chart here shows chlorophyll A as a solid line, chlorophyll B as the dashed line, and beta carotene, a type of carotenoid, as the dotted line. And so if you look at chlorophyll A, solid line, it peaks here in like the blues, and then it drops really quickly at the greens and stays basically at 0. And this is the what light is absorbing. And then it comes back up at orange and yellow, and then peaks again at red and kind of drops back down. Chlorophyll B is shifted over a little bit. So again, dark blues to really dark greens, plummets again down at greens, stays to 0, and then peaks a little bit at red again. And then beta carotene covers basically all the blues and purples, and the green goes to most of the greens, and then drops back down and doesn't come back. Now, the color that you see from a pigment is the color that it's not absorbing, right? And so you can see here with the greens, none of them really absorb too much green, which is why plants look green, because that's not the color they're absorbing. It's the color that's being allowed to reflect back to our eyes, so we see them as green. Now, the reds here and the oranges are only being absorbed by the chlorophyll A and B, not by the beta carotene. So oftentimes the beta carotenes will appear to be reddish or orange to us because they're not absorbing those, which is why the colors of fruits and seeds and stuff are often from carotenoids, because they're not absorbing light in those spectrums. They're reflecting it back to us. So we have chlorophylls that are green, at least to us, they show green. Carotenoids are usually yellow, orange, or red. And then anthocyanins that can kind of vary. They can be red, purple, or blue. And the cool thing about anthocyanins is that their color depends on their pH. And so, depending on which pH they are, their color will change. Acidic will be different from basic, and neutral will be different from both. Chlorophylls absorb red and blue lights. Carotenoids absorb blue, green to blue lights, what we just talked about there, right? Chlorophyll's job is for photosynthesis. Carotenoids is to assist in photosynthesis, to protect it, but also to color different parts of it. And then anthocyanins give coloration of fruits, flowers, different pollinators, things like that. There's some examples there. So we talked about chlorophyll A and B. We talked about beta carotene. There's also xanthophylls that are a carotenoid that give a lot of yellow pigmentation. And there's a lot of different pigments we're not talking about here. These are just kind of like the main ones. In the anthocyanins, there's one called cyanidin. This is what gives blackberries their color, why they're so dark. And anthocyanins serve mostly a coloration function, while carotenoids and chlorophylls chlorophylls are main pigment of photosynthesis, and carotenoids also help with that, while anthocyanins are less involved with that process. Oop. Okay, so we're going to start getting into the light-dependent reaction, so the first part of photosynthesis. Now, I'm going to use a different diagram to explain it to you, but I think this kind of shows that at least a cascade here. So we're going to take light, raise the energy of an electron, let it cascade down in energy to another photosystem, re-energize the electron, bring it back up to high energy again, and then let it cascade back down. So we're basically using light to excite an electron, making the electron really high energy, then letting the energy drop and trying to capture that energy, and then excite it again, and then letting the energy drop and try and capture that energy. And that's going to happen through two different photosystems, so photosystem 2 and 1. Photosystem 2 comes first, then photosystem 1. Between those, we have an electron transport chain, very similar to like we had in cellular respiration, with the goal of pumping hydrogens across. After photosystem 1, we have another electron transport chain, but the goal here is to produce NADHs, not to pump hydrogen. Okay, so let's kind of take a look at this. Here is a diagram of the light-dependent reaction. Here is a thylakoid interior, and the thylakoid membrane. Here's photosystem 2. Here's phytoquinine and cytochrome. Here's photosystem 1, and here's the ferredoxin complex. So what's going to happen here is light will strike photosystem 2. So light's going to go through the leaf, hit the photosystem 2, and it's going to excite the chlorophylls. Now, at this point, there is no conversion yet to chemical energy. Remember, we want to take light energy, photons, and convert them to chemical energy. So in the form of molecules, and we haven't been able to do that yet here. So light hits the chlorophylls. They get really excited and they start to transfer electrons, or not electrons, energy around. So you can see that here. Light hits this chlorophyll, these darker green circles, and it transfers the energy around. It's going to transfer it to the photosystem 2 reaction center. And inside of there are 2 chlorophylls. You don't have to know the name of them. There are some special names to them, but you don't have to know that. And what they do is they get really excited and they release an electron. So they release an electron, and an electron's going to go transfer down the electron transport chain over here. But it does not like to be missing that electron. And so what the photosystem 2 does is it takes water that has been transported from the roots to the xylem up to where the photosynthetic tissues are, and it splits it. And it splits it and captures the electrons. So it captures 2 electrons from water, splits it into hydrogen and oxygen. That hydrogen is going to be stored in the thylakoid interior here. The oxygen it produces is part of that equation, right? It's going to produce excess oxygen, which it can go to cellular respiration, but I think it's about 10% of the oxygen it produces goes to cellular respiration. The rest of it wants to get rid of. And so it's going to try and let it diffuse out of the stomata. So oxygen production and hydrogen production. Hydrogen is being stored, get rid of the oxygen. So we have that electron. The electron's been transported around. It's going down the electron transport chain. It's going to go to this mobile electron carrier called phytoquinine or plastoquinine. And it's going to take the electron over here to the cytochrome. And it's going to do the same thing that happened in the electron transport chain of cellular respiration. It's pumping hydrogens across. So it's taking hydrogens from the stroma and it's transporting them into the thylakoid interior. You can see that here, right? So it transports it into the thylakoid interior. So we're building up a reserve of hydrogens inside the thylakoid. So the goal of photosystem 2 in the electron transport chain is to take hydrogens from the stroma and put them into the interior of the thylakoid. That, coupling with the breaking of water, creates a lot, and the release of hydrogens from that water, create a lot of hydrogens in that internal space. Now, that electron's not done though, but it has dropped down energy. So if we go back to this diagram, we've now had the light hit here, we've excited the electron, we've broken the water over here to replace it, we've gone down here's the plastoquinine, we've gone down the electron transport chain to pump hydrogens, right? Now it's going to go to photosystem 1. And notice over here is energy. So low energy, excited, and the energy, as we pump hydrogens across, the energy's gone down. We need that energy back up. So photosystem 2 is going to accept light again. So here for photosystem 2, light hits another chlorophyll, gets passed down, and it gets attached to that electron. So that electron becomes very excited again, really high energy, right? And it's going to get passed down another electron transport chain where ferredoxin becomes one of the main molecules here. And the goal here is not to pump hydrogens. It's to take NADP plus and hydrogens on the outside and to create NADPH, another high energy electron carrier, just like we saw NADH inside of the electron transport chain of cellular respiration. So the goal of photosynthesis, photosystem 2, pump hydrogens across, break water, create a high concentration of hydrogens inside the thylakoid interior. The goal of photosystem 1 is to create NADPHs to transport that energy in the form of a high energy electron carrier to a later process. Okay, after that, the electron's not needed anymore. But we still have this reserve of hydrogens inside of here, right? And remember, things are going to diffuse from high concentrations to low concentrations. So we've made this high concentration of hydrogens inside the lumen of the thylakoid. And here we have another ATP synthase, just like we had in the electron transport chain of cellular respiration. Again, the hydrogens are going to flow down from high concentration to low concentration. And just like a hydroelectric dam, they're going to spin a turbine basically. And spinning that turbine from the process of diffusion starts to produce energy. So the ATP synthase takes adenosine diphosphate, and basically a spent ATP, plus the free phosphate group, and produces ATP. So it produces ATP here, as a product of the hydrogen concentration gradient. Photosystem 1 created NADPH. Photosystem 2 produces the oxygen and the hydrogen. That's the light-dependent reaction. That's this whole thing here, right? Light hits it, electron gets excited, replace the electron with water, go down the electron transport chain, pump hydrogens, excited electron again, gets really excited, goes down the electron transport chain, takes NADP plus H to make NADPH. So we have electron carrier, energy in the form of ATP, oxygen, and hydrogen ions. That's the light-dependent reaction. Oop. So now we have the light-independent reaction. So the goal here is to take the ATP and NADPH from the light-independent reaction and create carbohydrates. Remember, the goal here is sugar production, not energy production. So light-dependent reaction created batteries in the form of ATP and NADPH. Those are not the goal. The goal is sugar. It's carbohydrates. So for the light-independent reaction, we're going to take those energy carriers and run them through another cyclical pattern, like in cellular respiration where we had the citric acid cycle or the Krebs cycle. The same thing happens here, but now it's the Calvin cycle. So the Calvin cycle is going to take in those energy sources and make sugars out of it. Now, for the Calvin cycle, we're going to go through some steps here in a second, but you're going to take in CO2. So remember, the plants are breathing in CO2, taking in that carbon dioxide. That's where that comes into play. Notice no CO2 here. So take in CO2 and make sugar. And we're going to use ATP, NADPH, and ATP along the process. Now, what's important here is to understand the enzyme Rubisco. Notice here we have this protein, RUBP. Rubisco is an enzyme that's going to take RUBP and ideally carbon dioxide and allow them to combine. So the goal here, remember, is to make sugars. So in the citric acid cycle and in cellular respiration, remember, we take the glucose and we break it, take the energy, break it, take the energy, break it, take the energy. That was the goal of cellular respiration. Photosynthesis is the opposite. It's take a carbon molecule, add more carbons, add more carbons, add more carbons until you have a carbohydrate. And so the goal of Rubisco is to add the CO2 to another protein called RUBP. What's interesting is Rubisco might be one of the most important proteins on Earth. It's definitely the most abundant protein on Earth. It's found in all the plant tissues, right? And we eat all the plant tissues. So it's super, super abundant. And its goal is to take inorganic carbon in the form of CO2 and convert it into an organic carbon molecule. So it's fixing it. It's fixing carbon for us, right? This is where we get CO2 out of the atmosphere. This is where we get our carbon molecules. It's the foundation of most ecosystems here. Now, we'll talk about this in the next video more, but Rubisco is also very prone to errors. So it has a high affinity for CO2. It wants to take CO2 and add it to molecules, but if oxygen gets too abundant, it will take oxygen and do it. And plants don't want that. It's called photorespiration. So the goal for most plants is to make sure O2 levels stay down and CO2 levels stay up. Hence why when I was talking about oxygen production from the light-dependent reaction, some of it goes to cellular respiration, about 10%, I think. The rest of it, the plant wants to get rid of. It doesn't want that O2 in there. It wants CO2 to be saturated around Rubisco, so it has a very efficient photosynthesis production of glucose. And we'll talk more about that later. Okay, so here's the light-independent reaction, otherwise called the Calvin cycle. So it starts up here. We get 3 CO2 that come in. The plants are getting the CO2 from the stomata, so it's diffusing into the plant. And here's RUBP. So here's the protein RUBP. Notice it has 1, 2, 3, 4, 5 carbons on it. Rubisco is going to take the CO2 and the RUBP and make 3 of this 6-chain protein, or 6 carbon molecule. 1, 2, 3, 4, 5, 6. And it produces 3 of those from the 3 RUBPs. Right? So it's basically 1 carbon to 1 RUBP. Creates 1 6-molecule thing. Now, that molecule there is unstable. It breaks right away. So it splits. in half, and you get a 3 PGA molecule. So 1, 2, 3 carbons. And since you had 3 RUBPs, 3 of these molecules after CO2, you get them split and you have 6 3 PGAs, right? Now, for each one of those, you have to use an ATP. So ATP that comes from the light dependent reaction is used here. This is the carbon fixation. So carbon becomes, I should back up. Stage 1 is carbon fixation. So it's taking carbon and putting it into an organic molecule. We've taken the CO2, it's now an organic molecule in this 3 PGA, right? Now, we then want to try and get it down to where we can make the glucose for it, right? So we're going to go through a reduction reaction here. And so it's going to take ATP, NADHs, so it's taking the energy from ATP from the light-dependent reaction. It's taking the NADPHs from the light-independent reaction, taking that energy, and turning these 3 PGA molecules into G3Ps. Now, what's important here is we had 3 CO2s. We turned those into 6 3 PGA molecules, right? 5 of these G3Ps that come from those 3 PGAs get recycled in order to regenerate these RUBPs up here. So that way the process can continue. Because if this RUBP is used up, then you can't continue with the Calvin cycle. And so it needs to regenerate those. And so for every 6 3 PGA molecules we make, we get 1 G3P molecule. The other 5 get recycled, right? Now, G3P has 3 carbons. Glucose has 6 carbons. Remember, C6H12O6, 1, 2, 1. That means in order for us to make glucose from G3P, you have to have 2 of them, which means you have to go through this process twice, right? In order to get 2 G3Ps. Once you have 2 G3Ps, the plant will combine those to make glucose, and that glucose production is done there. Now, once you have these 5 G3Ps remaining, you use more energy from the light-dependent reaction in the form of ATP to regenerate RUBP, so you can continue down the process. So you can see it's quite a convoluted process. Combine the carbons, the carbons will break down, send one of them away in the form of a G3P, take the others and recycle the process, taking a lot of energy along the way. So that's the light-independent reaction. The goal there is to take in the CO2, which the plant is bringing through the stomata, make G3P. You need 2 of them to make a glucose molecule. So now we've talked about oxygen production, which is from the breaking of water in the photosystem 2. We've talked about hydrogen ion pumping from the light-dependent reaction to produce ATP. Photosystem 1 takes in light, right, and then makes NADHs. Those ATPs from photosystem 2 and the NADHs from photosystem 1 get transferred over to the light-independent reaction, where we take in the CO2 and go through a couple cycles to make the glucose. So we've now covered the entire photosynthesis equation there. So to recap, you should know photosynthesis, the discovery, the idea of that, the light-dependent, light-independent reactions, the role of rubisco, pigments, and light energy.